Hi, everyone. Hi. Next we have Orazio Marine. He is the stewardship specialist with the New York State Office of Parks, Recreation, and Historic Preservation in the Division of Environmental Stewardship and Planning. Orazio Marine is a tick and deer management specialist from the New York State Office of Parks, Recreation, and Historic Preservation. His surveillance has been able to capture the spread and establishment of the invasive longhorn tick in some monitoring state parks. Today, he's giving a presentation on Asian longhorn ticks, tick surveillance, and safety. Please extend a warm welcome to Orazio Marine.
Hey, how's it going, everybody? Thank you for the introduction, and I'm just going to jump right into it. So, I'll be talking about the Asian longhorn tick. I might use ALT or Hemophysalis longicornis interchangeably throughout this. That's the species that we're going to be talking about today. So, its native range is Eastern China, Japan, Korea, Russian Far East, and it is now established in Australia, New Zealand, several Pacific islands, and the United States. This picture is from USDA Agricultural Research Service. I reached out to this guy who is, at the time, he was working on looking for potential biocontrols. They weren't able to find anything yet, but they're hopeful that they might find some parasitoid wasps or entomopathogenic fungi. So, as you can see by this map, the preferred habitat is a lot of the eastern United States. They like temperate climates with moderate temperatures, humidity, and precipitation. One study found that they were more abundant in open fields and ecotone areas, and then as they got into June and July, transitioned to more wooded areas. So they do like this sort of patchy edge space where they can move back and forth between much of the eastern United States is suitable habitat. And as you can see, Long Island is very much in the red. So host preferences. It's a generalist three-host tick, like much of the ticks that we deal with out here. It's found on cattle, horses, sheep, goats, deer, foxes, raccoons, rabbits, birds, domestic animals, and can be found on humans. Migratory birds are very important when it comes down to this tick because they're able to transport it far distances. So they're, linked with sort of the moving fronts of where these ticks can be found. Asian longhorn ticks, fortunately, this might be one of the most positive takeaways that I have to share. Uh, they show a lower preference to human and paramyscus mouse hair. Paramyscus mice, that includes, the white-footed mouse, which is one of the reservoir hosts for a lot of our tick-borne illnesses. So having a lower preference for that and for humans might be good for us. But they definitely host on domestic dogs, cats, and white-tailed deer. Infestations can become very severe in agricultural settings. You can run into problems with growth and production. You're going to get economic losses, and animals can be killed by exsanguination as well. So its importance as a vector. It's a known vector of Thaleria orientalis Akita strain, which is a protozoan parasite that causes bovine infectious anemia. This has been found in the United States and New York. It can carry a variety of pathogens, including anaplasma, Ehrlichia, Babesia, severe fever with thrombocytopenia syndrome virus, Russian spring summer encephalitis, and Powassan virus. Genetic material from Anaplasma, Babesia, Ehrlichia, and Borrelia burgdorferi, the Lyme disease pathogen, and bourbon virus have been found in field collected specimens in the United States, but their actual ability to transmit those remains largely unclear. Lab studies have found that ALTs are not competent vectors of Lyme disease and tularemia. Those pathogens don't survive through the molting process. Um, but it can transmit the causative agents of Rocky Mountain spotted fever, Heartland virus, Bourbon virus, and Powassan. So some unique characteristics about this tick. We mentioned before that this tick, the Lyme disease pathogen and some of these other tick-borne illnesses don't survive the molting process. Um, Something that's interesting about that tick that kind of complicates that situation is that they are found sometimes host seeking while partially fed. I've run into a number of them like this. These are actually pictures that I took with Moses Asura of Suffolk County Vector Control. I collected those ticks from Robert Moses State Park. Some of them were definitely host seeking while partially fed. I haven't seen that in other species of ticks yet in my samples. And it's reflected in the literature too. People tend to find them like this. So that kind of complicates that situation and leaves us with the question, can this tick transmit disease from that partial blood meal to the secondary blood meal that it collects if it does so? Now, I certainly won't be volunteering for that, but it's a question that we should try to answer. Um, they're parthenogenic. That's the big one with these. That's part of the reason why migratory birds can play such a big role in moving them around, because if they are successful with their blood meal as an adult, they can produce up to 2,000 eggs without ever having to find another Asian longhorn tick to breed with. So it really only takes one to get that population going as long as it's successful and the larvae are successful afterwards. And, 2,000 larvae, they have a pretty decent shot at getting to that next life stage, at least a few of them. Um, they have been found to be able to complete one generation in a year. This is from New Zealand. Our native tick species are generally 2 to 3 year life cycle. In ideal conditions, they can get through a generation in as little as 6 months, probably more within agricultural settings and indoor, settings like that. They do go into diapause just like our native ticks, so they're not going to be super active in the winter. Um, but they can get through those generations a little bit faster. Might have something to do with the fact that they're parthenogenic and never have to go through that mating phase. So these are some of the key ID features. On the left, we have Hemiphysalis leporus palustris, which is the rabbit tick. That's actually a native tick in this genus that we have that's almost ubiquitous in the United States. It's very closely associated with rabbit hosts and burrows, so it's very uncommon to find them during a flagging survey. I have found them. Uh, Kind of freaked us all out for a second, but we were able to confirm that they were Leporus palustris and not longicornis. So you can see on these um, members of this genus have these palpal segments that kind of extend a little bit farther out from the basis capitulum. Most of our native ticks are pretty straight when it comes down to those palps. But these guys have sort of like a wire cutter or wrench shape to them. Um, so those features right there, that's the flared palpal segments I was talking about. They go out past the basis capitulum, kind of think like the neck of the tick. Um, and those ventral spurs, this is where I've come to understand is where they get the horn in longhorn tick. It looks more like a fang to me, but the Leporus palustris does not have that super pronounced fang-like structure. So that's something that you can use to compare between the two if you're trying to figure out which one you have. And I also tend to think of Leporus palustris as having much more triangular shaped palps. They're very much wire cutter shaped versus the Asian longhorn tick, which has the more wrench or pliers shape to them. I did hand out some guides. Feel free to take those home. If anybody wants more copies, I'd be happy to share them. So this is just to give you a comparison of larvae. On the left, we have the Asian longhorn tick larva. They have that wrench shape going on, that sort of pliers shape. And then if you look to the right, you have Leporus palustris, very pointy, triangular. So just, if you're worried about it, try and look to that shape. Structure is very important when it comes down to identifying these ticks. So coloration is not always something that you should rely on when you're looking at what kind of tick you might have. These are two uh, uh, Asian longhorn ticks right next to each other. You can see there's a difference in pigmentation. There's another picture of the same two ticks. I just kind of zoomed out a little bit. You can see the coloration is definitely not the same. So you got to watch out for that. On the right, we have two adult male Lone Star ticks. You could see the patterning is different on them. A lot of people think, Lone Star tick, it's the tick with the white dot in the center of the back. The adult males don't have that feature at all. So it's very important to look at structure. I generally look at the shape when it comes to Lone Star ticks. Very rounded, almost crab-like, very, very long palps, mouth parts going on with them. All right, so the story so far. Uh, this detection first happened in 2017 in New Jersey. Um, I believe it was on a sheep. I'm not 100% on that. And they looked back into their records at some of the earlier submissions that just were never ID'd to species, and they think that it arrived probably sometime around 2010 or before then. I believe the earliest record that they have is from 2013, and that was something that they had to sort of go back and look at specimens that were submitted earlier on. So the spread has been pretty rapid throughout the United States. We're pretty much it's 2025, going to be 2026 soon, and we had them in just New Jersey, and look how far they got. Robert Moses State Park was the first internally surveyed New York State Park. I say that because I don't know if somebody else found them before I did. Um, but they first showed up in my survey results back in 2020 at Robert Moses, and very quickly sort of started to show establishment. Um, so the most recent occurrences, I'm in a working group that meets in the wintertime, it's once every other month, but for the rest of the year, it's usually once a month. And we just kind of provide each other with updates on where the situation is across the US. It showed up just this year. Since April 2nd, we now have them found in Berrien County, Michigan, Van Buren County, Iowa, Cumberland County, Maine, and Franklin County in Kansas. So it's continuing to move. This is a nice trend, but it's not what we really are happy to see. Um, so I first encountered the tick in 2020 at Robert Moses. That year, we weren't identifying nymphs to species. It was just the adults. I advocated that nymph ID was important to do, and we started doing that the following year. Um, so this is a chart of the relative abundance of Asian longhorn nymphs and adults compared to the other species that we were finding in the park. And we started off 2021, we were at 22% of the nymphs being ID'd as Hemiphysalis longicornis. And now 2025, this is just between April and July of each year. 2025, we're at almost 83% of the nymphs. We didn't really see this pattern too clearly with the adults. I'll say that these 2 years, very, very big dog tick years, for whatever reason, we were finding loads of them. So that might be why that's kind of muted for those 2 years. But yeah, this is the relative abundance pattern that we've seen. So very quickly, they've established dominance. In the other parks, we see not as severe yet. Wildwood, They actually first showed up in Wildwood as larvae in 2022. I was sampling the maintenance yard, and I noticed that the the larvae that appeared on my flag were a little bit darker and more opaque. I know I was just saying before, don't rely on color, but something that I've noticed with the Longicornis larva is that they're a little bit darker, a little bit more opaque, and even a little bit larger than our Lone Star larvae that we usually run into. Deer tick larvae tend to be grayish, blackish, and even a little bit more transparent. Um, so I saw these larvae. I thought they were suspicious. I looked at them under the microscope. They turned out to be Longicornis. I do not look at every larva that we collect under the microscope. Um, that would be crazy. Uh, but this is at Heckscher as well. And then Connetquot, not as bad. Hopefully doesn't get as bad as Robert Moses. But it looks like Wildwood might wind up getting close to there. One thing I think is interesting is that uh, Wildwood and Heckscher are other coastal parks. I don't know if that has something to do with shorebirds moving these things around, but it's something to think about. Um, so yeah, uh, this is not just observations that I've made alone. It's available in the literature as well. Back in 2018, there were two very extensive tick surveys that were conducted on Long Island, and Longicornis was not collected during those. But there were, I'm not sure how many individuals were found. One survey that was done by the Suffolk County Arthropod-Borne Disease Lab, uh, they did find Longicornis from a sample site in 2018. I believe it was somewhere on Jones Beach Island, either between Jones Beach and Gilgo, somewhere around there. That was what they had circled on their map. Um, by 2022, which was when a follow-up survey was done by this Rockland et al. 2023 research, uh, Longicornis was dominant or principal tick species in many of these areas. You can see on these little pie charts, the stars are some of the collection sites that occurred during one of those 2018 surveys where Longicornis was not found. And right nearby, just a few years later, 4 years later, they have dominance in areas that were sampled that were very, very close to where those previous areas were surveyed. So it seems like they can get their numbers up very quickly. Again, that parthenogenic aspect of it, very advantageous evolutionary strategy on their part. Um, Other tick species, deer ticks, Ixodes scapularis and Amblyomma americanum, the Lone Star tick, they've taken decades to expand and become dominant. Uh, Lone Stars have been cited as first being detected on Long Island in the 1970s, expanding through the 90s. I remember when I was a teenager, I never encountered them really. And now we all work in this field. We know that you don't want to go outside in August with shorts on sometimes. So, I hope I'm preaching to the choir on that one. Um, so why does New York State Parks do tick surveillance? Um, we started doing this because it's a reporting requirement for our four-poster permits. It's a deer treatment station. Deer come in, they're attracted to a bin that has a corn dispenser, and during that, they come into contact with permethrin-treated rollers, which treat the deer on the necks and ears with permethrin, which helps target the ticks that the deer are carrying. It's intended to the surveillance is intended to monitor the efficacy of this with sampling areas situated inside and outside of the treatment zones. Um, the treatment zone is kind of a rough, I believe, 40 acres around each four-poster, but we know that the true area of effect is actually determined by the movement of the deer themselves, where they were before and where they go after visiting a four-poster. Um, So another great thing about the surveillance, not only looking into the efficacy of the control methods that we're using, but because we're doing this surveillance, we're able to capture these new introductions. We can see seasonal activity and we record abiotic factors with every sample that we take. So we're getting microclimate data that's connected directly with the tick activity that we see from that sample. Um, and yeah, really the surveillance to be able to capture these new introductions, everyone knows the saying, if a tree falls in the woods and nobody hears it, you know, it's kind of like that. If you're not looking, you're not gonna see anything. So it's good to keep your eyes out. So the surveillance method that we use is the flagging method. We take a corduroy flag and drag it through set distances in our tick sampling areas. Um, we sample either transect areas or grid areas, depending on what the available habitat is. We can't really lay a grid out in the dunes at Robert Moses because we'll be dragging over sand for a lot of it. So we need to be able to create these meandering transects in some areas. Um, the sample size is a standard 55.5 meter sample. That was the number that I was given and told to do. I know it's a little bit of a weird number, but that's what we use. Uh, we also collect, like I said, the microclimate data with each sample by a Kestrel weather meter. We do this bi-weekly weather dependent. You don't want to be going out and sampling when it's raining, if there's high winds. You're just not going to be able to capture the tick activity if it's like that. Generally, we try and go out if the overnight temperatures were 40 or above, we usually try and stick with that. Um, that said, I have seen and run into ticks while there was frost on the ground in the morning. By the afternoon, you could be finding ticks on yourself. So, winter time is still very much an active season for our native deer ticks. So, um, for the collection techniques, I'm sure a lot of us are familiar with this kind of site, usually beginning in August and ending sometime in October. Larva season, always fun. We remove the ticks from the flag with a lint roller. It's a great tool not only for your own protection, but for the sampling itself. Um, we like to I roll up the sheets. So what I'll do is once I collect my specimens, I'll make a little fold on the first part and then actually roll the sheet up so that way it doesn't stick to itself and then you can pull it back out and examine it later on. Um, so we take the rolled up sheets and we put them inside centrifuge tubes, which get frozen. We mark them with the location, date, and date information, what transects were sampled, and we freeze them before we take them back out to ID and count them. So that's what they look like. You take a Sharpie, you write your information on there, and freeze it up and take a look at it later. All right, let's talk about fashion. Uh, One thing that if you're going to be doing surveys like this, you want to make sure that you're keeping your items hands-free but accessible, especially when you run into a bunch of tick larvae. You don't want to be floundering for what you need to get while the ticks are spreading throughout the flag, making themselves harder and harder to collect. So I really like to use a fanny pack with a water bottle holder. This thing's a little beat up. It's seen 6 years of tick surveys out there. So, Amazon does have stuff like this, wherever you can find that little water bottle holder is great as a little lint roller holster for you. Um, so I keep my specimen tubes and my lint roller in that with my Sharpie. The shoulder bag's good for your clipboard, data sheets, empty specimen tubes, and you got to be wearing PPE. I use permethrin treated coveralls. I know a lot of people will use Tyvek, but the more you wear that, it might not last a whole season of repeated use, So something a little bit sturdier, some coveralls, you get those treated with permethrin. Uh, that can be effective as well. I really lean towards the PVC boots because they don't have any hiding places. When you have your hiking boots on, you still have a crevice between the tongue and the laces and everything like that. When I first started doing this, I would wear my work boots and I would come home and tap my boots out and ticks were falling out. So you could track them around. Don't don't bring yourself home any surprises. Uh, during larva season, really helpful. I'm sure a lot of us already practice this. You put that few wraps of duct tape out around where the boot line meets the pants or the coveralls, really helps them from not, helps prevent them from getting in. So you definitely want to try to, oh yeah, I don't know if I'm going to have that last bullet under there. I can't remember what I put there, but wear sunglasses. If you're out in a field looking at a white flag that's covered in ticks in the sun, it's not going to be pleasant. So it's definitely good to have sunglasses on if you can. Um, when it comes down to just the general principles of tick safety and prevention, you want to reduce those entry points and hiding spaces, like I was saying about the boots before. Use your repellents and treatments of your best judgment. Always read the label. Um, Funnel them up and out. The ticks are naturally inclined to go up. So the more you keep funneling them to the outsides of your clothes and keep them moving up, so that way they're going to be continually, climbing over hopefully permethrin treated garment or something like that. And sometimes you might even just brush against something and knock them off. As long as they're on the outside of your clothes, they're not getting to your skin. Try and keep them off your skin for as long as possible. Make it hard for them. Check with a lint roller very frequently, especially before you get back into your car after you're doing survey work. I like to keep them in the door jambs. That's generally what we do. That little door pocket, have your lint rollers in there. It's like a visual reminder to do the check as well. Avoid dense vegetation and leaf litter. Most of us will know what tick habitat is. They really like the leaf litter. So just be mindful if you're going into tick habitat, you want to take these steps to protect yourself. And be vigilant when the humidity is high and the winds are low. I think if it's bad mosquito weather, it's probably going to be bad tick weather too. They kind of they're active during those times. So uh, dehydration or desiccation is one of the most dangerous things that a tick has to worry about. So if you have low winds and high humidity, they're going to be able to spend more time questing. So your odds of encountering them are going to be greater. Those are the coveralls I was talking about. I like to use speed suits, I believe they're called. You have to be careful if you're going to get coveralls that they don't have the opening for the access to your inside pockets. Worst comes to worst, you can always take a sewing machine, sew that up. But the speed suits don't have. And they're short sleeve, nice and comfy in the summer. I get my boots from Dunlop. That's the rhino skin. I think that was the last thing that I had on that the other slide. So it's kind of like a tight fitting shirt. They make leggings and socks too. I like to use that in the springtime during deer tick nymph season. Um, it's just another layer of defense. There have been times I've come home, found a deer tick nymph like right on the outside of my arm, and I was like, you got me. So that can definitely help prevent stuff like that from happening. The deer tick nymphs are really, really small, and you don't really ever feel them on you. So it's good to have an extra layer of defense, especially that time of year. Uh, I like to use the permethrin treatments on most of the clothes that I wear. Sometimes I'll send my stuff into Insect Shield to get that treated. They'll do the coverall treatment for you. Um, uh, I know a lot of us might use the aerosols. Uh, whatever your preference is on permethrin. Obviously, always read the label. Use your best judgment on what products you want to use. I lean towards the squirt bottle because I don't like when the aerosol can gets all spittly at the end. Um, and obviously lint roller. Lint roller is going to be your best friend, right? Do checks often. Don't worry about wasting the lint roller sheets. Just keep using them. All right, so one of the last slides that we have here, safe removal. The main principles of this will be you want to remove the tick by the head or the mouth parts with a firm but controlled motion. Try not to break it, try not to squeeze it, and try not to agitate it. Um, the more that you kind of disturb the tick and mess with it, the more chances you're going to have that it could regurgitate something back into you. And that's where you're going to run into problems. If you're going to, if it's good to clean the wound and everything like that, but don't do it while the tick is still on you. That's going to agitate the tick for sure. Definitely clean it, but wait until after you get the tick out. Uh, Tweezers, with them, you want to grab by the head or mouth parts. There's another thing called a tick key or an easy tick remover. I've got some pictures of these. Um, so obviously the tweezers, you want to be grabbing by the head. The tick key, I actually have one. You can kind of keep them as a keychain. It's got a little notch. So you slide that over the tick and it can help pop them out, you know. When it comes down to like my dogs or if I have a tick somewhere in my hair, I really like to lean towards those easy remover. Looks like a little crowbar, but it helps get past the hair and everything like that without ripping it all out. So, uh, great on pets. But so the don't cover a tick with petroleum jelly or Vaseline. Don't burn them with a match or a lighter or a cigarette. Any volatile substances like alcohol, gasoline. I've read specifically don't use kerosene. Hopefully we don't do that or but yeah, definitely don't do that. And try not to pull them off by hand. If you can get a good tool, you really don't want to squeeze the tick. Um, yeah, that's going to increase your risk of disease transmission. I just want to preface for say this at the end. I'm not a doctor. Talk to your doctor about these things too. But these are just some tips. And if you do have a tick, there's a website. They used to do it for free, but their funding kind of uh, didn't hold up over the years. But for around $20 to $80, depending on how and thorough you want them to be, you could send a tick to nyticks.org and they'll do a lab test on it for you, see what it might have. Again, a conversation to have with your doctor. One last thing. I hope I didn't fly through that too fast, but this is my last slide. I just kind of an honorable mention here. The Gulf Coast tick, Amblyoma maculatum. That's going to be coming soon, probably. We have established populations in New Jersey, Connecticut, and Staten Island. Uh, So far, we've found 10 confirmed adult specimens on Long Island since 2021. Uh, I've been sort of sharing information on all this with Suffolk County Arthropod-Borne Disease Lab, Suffolk County Vector Control, and New York State Parks. Uh, so anytime any of us find that, we're all collaborating with each other, letting us know. We have a little map that we've been sharing of the locations. Um, in terms of the state park tick surveillance that I do, I found one adult male first in 2022. And then this year, in July, I found an adult female um, at Robert Moses as well. Robert Moses, they got them all. Uh, you guys actually beat me to it and found what I understand is the first one on Long Island back in 2021 at Sayville Grassland. So thank you. Um, and it is a vector of Rickettsia parkeri and Hepatozoon americanum. So Rickettsia parkeri, it's another form of spotted fever. That's what we would have to worry about with them as humans. And then Hepatozoon americanum causes canine hepatozoonosis. What's interesting about that is that uh, a dog can get sick from it when if they're grooming themselves and eat the tick, rather than the tick transmitting it through, biting the dog. So it's like the other way around. It's weird. Um, but from what I've read, it's occurs at very low numbers in the actual environment. It's kind of rare to find that. Hopefully it stays like that. Um, but yeah, I don't know if I was a little too fast on that, but these are my references and so our four poster program is happening at Wildwood State Park, Heckscher State Park, Connecticut. Oh, the question was where do we have the four poster program implemented in the Long Island region? I believe Clay Pits in New York City region also has a four poster. I don't know if they still have it, but the last I heard, they were doing it as well.
Any correlation with like the densities of these ticks and any specific plants or vegetations? Like, I know, for example, like Japanese barberry has a correlation for having a lot more ticks. Is that like just ticks in general or is that like specific to a certain type of tick?
Great question. The question was, have I seen any correlations between the habitat types and vegetation and the densities of the Asian longhorn ticks? Um, Generally, what I've seen is that, obviously Robert Moses has the most severe infestation so far. Very much a open shrubland, not certainly not a closed canopy, forest understory situation at all. Um, the literature, they do tend to say that it's these field areas next to like the sort of those ecotone areas. In one of the studies that was actually going on in upstate New York and or Westchester region, I believe, they were finding them on mowed grass. So they do tend to be a more like open sun, like a little braver and going out into the open sun than some of our native tick species. I've heard from the Asian Longhorn working group that somebody else who was also in a sort of maritime dune shrubland area had a similar situation where the uh, uh, Asian Longhorn ticks infested very quickly and like quickly started to show signs of becoming the dominant tick in the area. So it does seem like more open sort of field-like areas, like they were saying. I believe it was May through June or April through June. They have a lot of density in those open field areas, and then they kind of transition to more shaded as it goes later into the season. Um, but it seems like these guys are a little bit more tolerant of that open environment. So that's my take on it. Thank you for that question. That's a great question. The question was, are they more questers or active hunters? I'm not aware of, I haven't actually looked into that. So I don't know if they're using that CO2 tracking like Lone Stars are. So I don't know, but I can get back to you. Oh yeah, on the roller themselves. Yeah. Sometimes they can kind of sit themselves back up. I've had this similar question. The question was, Are the ticks getting stronger or are the lint rollers getting weaker? I, I think the lint rollers are getting weaker. I hope. So the main one is going to be for the agricultural setting, the Thaleria orientalis Akita strain. That can be a serious problem if you're a cattle farmer or dairy farmer. So that's something to look out for. They in laboratory settings, I believe, let me flip back over here. In laboratory settings, they were able to transmit Rocky Mountain and a few other pathogens, but for the most part, they think that they can't be a competent vector for Lyme disease. However, we talked about the partial blood meal aspect of things, where it's actually going for a secondary blood meal after hosting on something else first. I think it remains kind of unclear as to how that changes the sort of threat of disease transmission. Uh, but we know in laboratory settings, we've found them with Heartland, bourbon virus, and Powassan virus, and I believe recently in Connecticut they found them with Ehrlichia schaffiensis. But still, there's a lot of unanswered questions as to what their vector potential is to humans. And one good takeaway is that they have a low preference for human beings. I've never been bit by one. I believe Suffolk County has one report of a human bite. It's pretty rare. It does happen, but it's pretty rare. So silver lining on the cloud there. Yeah, yeah. On me, I don't know. I know that I believe the sort of record for larva, one sampling area, 155.5 meter sample, over 3,000 larvae that I collected. That was at Wildwood. That was at Wildwood, but they're actually doing better than a lot of the other parks. There was just like a freak, thing that happened. Um, I'm not sure from eating an infected animal. Oh, I don't believe so, but I actually I haven't looked into that. Yeah, that's a good. Um, yeah, not that I'm aware of, but it's not something that I've done too much research into. But anybody else? All right, awesome. Thank you so much, guys.
Note that our next meeting will be all virtual, so you can enjoy Zooming in with us from home on February 24th. And also remember to save the date for our Resilient Long Island Symposium, which will be a little bit after that. Also, it's a Tuesday, not a Friday, for scheduling reasons. So save the date and join us on Zoom all together. And thank you so much.